Hello, everyone. Welcome to today's webinar by the Global Antibiotic Research and Development Partnership, GARDP. My name is Victor Kwasi, and I'll be hosting today's session on exploring non-traditional antimicrobials, insights from 3 cases. Revive is GARDP's education and outreach program. It aims to connect and support the antimicrobial R&D community by facilitating learning, sharing knowledge, and connecting people. These webinars are part of our educational activities. All of our webinars are recorded and freely available to view after the live session on our website, revive.gardp.org/webinars. You can also read our series of articles known as Antimicrobial Viewpoints, where experts discuss various topics within the field. And we also have a resources section that includes the Antimicrobial Encyclopedia. Here you can see definitions on various important terms that we mentioned here in the webinar. And some of these also include videos where experts give a further explanation on the terms. As always, today's presentations will be followed by a Q&A session. You can submit your questions at any time during the webinar via the questions window, as shown on this slide. We will address the questions after the presentation and do our best to respond to as many questions as possible. If a question is for a specific speaker, please do indicate their name at the beginning of your question. Today's speakers are Jennifer Schneider, Rida Mourtada, and Gregorio Iraola. Our moderator is Sina Gerbach. Sina Gerbach is the Deputy Head and Development Lead of the Transfer Group Anti-infectives, Leibniz Institute of Natural Products Research in Infection Biology. She's also Program Manager at INCATE. Her interdisciplinary expertise in project planning, portfolio, and stakeholder management developed through several positions in academic research consortia, but also in drug development programs within private-public partnerships. She holds a PhD in cell biology from the University of Dresden, Germany, and completed postdoctoral positions at Uppsala University, Sweden, and at the Max Planck Institute for Molecular Physiology in Dortmund, Germany. Welcome, Sina. I now hand over to you.
Thank you, Victor, for the nice introduction, and thanks to GARDP for this initiative and a really good collaboration. And hello, everyone. I take this opportunity to say a few words about INCATE, the incubator for antibacterial therapies in Europe, which is actually a Swiss-German public-private partnership supporting early startups or projects that are still academic but are about to spin off. And we cover the field of antimicrobial resistance. And what INCATE provides is advice, both scientific but also business development advice. INCATE also provides community, meaning we connect you to experts that you may need. We introduce you to the network. And INCATE also provides funding, which is non-dilutive. But advice and community is independent of the funding. So applications are actually accepted from around the world and continuously, so no deadlines. Just drop us an email, a message, or a call whenever you have an innovative project. But let's start now with our today's webinar, and I welcome our first speaker. This is Jennifer Schneider. She is a CEO of Centauri Therapeutics in the UK since May 2022, and she has almost 20 years of experience in antibiotic policy, in partnering and fundraising. She worked with companies, but also with universities and NGOs in the US and worldwide. And her topics are market creation, access, and novel payment models. And I'm looking forward to your talk now. Welcome.
Thank you. Great. And first of all, thank you to GARDP for including Centauri in this conversation today. We appreciate the role that GARDP has in the antibiotic community and how valuable Revive is in supporting the whole ecosystem. As mentioned, Centauri is based in the UK. We're up at Alderley Park, which is in the northwest of the country, and we're about a 12-person team with most of the team up in the labs in Alderley Park. I'm excited to share with you our work to develop an immunotherapy to address infections in the most vulnerable patients. So given that the focus of the webinar today is on non-traditionals, I wanted to start out with why we think there's a need for new modalities to address infections, which goes beyond what we typically think of as resistance. So yes, we know that there's a huge issue with resistance. We think about different resistance mechanisms, but beyond that, we think that's just the tip of the iceberg in terms of the infections that need a new way of being addressed. So for example, in 2023, there were 3 phase 3 readouts. And in those studies where people had been screened for resistance, there was between a 35 and a 50% failure rate in those studies. And that's both for the drug that was being studied, as well as for the standard of care. And this is coupled to all the other data that we have in other patients, like cancer patients, transplant patients, individuals who are taking chronic antibodies, folks on steroids. So we think the pot of people out there who need a different way to address infections is actually quite large. And we think this is the role of a novel modality like what we're developing. So this challenge was clearly described by Dr. Steve Holland from the NIH at the recent ESKAPE meeting in Barcelona, where he stated, we've all been there. We have the bug, we have the sensitivities, we're giving the right antibiotics, the patient is even taking them, and they are not getting better. And we think that this is the call, the role, the need for non-traditionals to meet this challenge. So we know that this traditional approach to culturing the infection from a patient, checking its sensitivities, and treating with said antibiotic is no longer sufficient, as this approach doesn't take into account any of the complexities of the host. We believe that by taking the host into account, the patient into account when developing drugs to treat bacterial infections, that there's a better opportunity to address those infections which are currently not being well treated by current modalities. And again, these are patients who are immune suppressed, elderly, on steroids, on antibody therapies, and we believe that these are our patients. And just to drive this point home, one of the top papers in antimicrobial pharmacology from 2023 also presented at the ESKAPE meeting noted, typically antimicrobial drugs are administered to assist a compromised or functioning immune system in combating an infection. However, the host response component is usually disregarded during PK/PD assessment of antibiotics, and its effect relative to that of the administered antibiotic is undetermined, although it may be profound. So our objective at Centauri is to use immune modulation to tip the balance back in favor of the host. We know that anti-infectives work best when coupled to a robust immune response. So most anti-infectives work, they come in, they drop down pathogen load, and the immune system comes to the table to clear up the rest of the infection. However, in patients such as those that I've been describing, the drug is still coming in and doing exactly what it's supposed to do, drop down pathogen load. But when the immune system can't come to the table and help clear infection, that's what leads to further morbidity, continued hospitalizations, potential for the development of resistance. So we think that by bringing the immune system back into the equation, we can address some of these more difficult to treat recalcitrant infections. So we think that immune therapy has the potential to transform anti-infectives in the same way that discovery of the cancer immunity cycle has for oncology. So just as with cancer patients, we think we can provide additional armamentarium. So as with cancer, things like chemotherapy and radiation still do a lot of the heavy lifting in treating those patients. And we see the same thing with antibiotics when we think about how a molecule like ours could be used. Traditional agents like the beta-lactams will continue to play a major role and do a lot of the heavy lifting. but there are infections and there are patients for whom these additional modalities will make a difference. And just a bit of background on the technology before getting into the detail. We call our platform Alphamers. What we're looking to do is recruit pre-existing natural antibodies that exist in everyone. They're ubiquitous, they're already perfused in all individuals, they're polyclonal, they're IgA, IgM, IgG, and we're looking to leverage multiple arms of the immune system. So just a quick shout out to our funders, because as we all know, to develop a drug, particularly in this space, requires a village. So huge thank you to both our private and public funders who've provided both financial as well as significant scientific support for us to take our programs forward. So the background of the platform and the technology. So the whole platform is based on a phenomena that was seen in the 1960s when physicians tried to use pig tissue for heart valve transplants. And while they were prepared to manage a graft versus host type response, they saw an almost immediate killing of that xenotransplant. And they realized that this killing came from antibodies that were already present in these patients for whom they tried to use this foreign tissue. So our platform is really, how do we take advantage of that antibody repertoire that already exists? And rather than kill off a xenotransplant, can we kill off a pathogen? One of the important things to note about these antibodies is, in this context, in the 1960s, one of the things they tried to do in order to overcome this antibody response was just continue to immune suppress these patients to try and knock out this repertoire. And they were never able to do it. And that's why we now actually have knockout pigs, right? Because this antibody repertoire is just really hard to get rid of, which is why we think that there's probably a huge spectrum of patients who have the necessary components of the immune system and these antibodies to respond to a drug like we're making. So the key to the antibody response that was seen in the context of xenotransplantation, and that we're also trying to take advantage of in the design of our molecules, is all about the presentation of natural sugars to the immune system. So let me get to my pointer. So in the context of xenotransplantation, those cells were largely coated with a sugar called alpha-gal. The antibodies were recruited because of the high density presentation of those sugars on the cell surface. And this is important because these antibodies are actually very low affinity, but they're high avidity. And I think of avidity like Velcro. You know, if you think about using a Velcro strap to tie your shoe on, if you just engage a corner of that strap, you're not going to have very good binding. But if you lay down that entire strap, you actually have really good engagement. And that's what the avidity effect is. So really, we're laying down our Velcro here to engage that antibody repertoire. As mentioned in the xenotransplant context, these sugars were largely alpha-gal sugars. But we know that the same mechanism exists whether you coat your cells with alpha-gal sugar or another sugar called rhamnose, and we have molecules which express both of those sugars, not at the same time, not on the same molecule. So the design of our molecules, on one end we have a targeting domain where we're able to selectively bind our targets of interest. In the case of our lead program, that is a gram-negative binder, and importantly, this targeting domain does have intrinsic antibacterial activity as part of its property. On the other end of the molecule is the effector domain, where we present either that alpha-gal or that rhamnose sugar to the immune system. And those two ends are coupled by proprietary linkers, which allow us to ensure that we're presenting that sugar to the immune system most appropriately. So this cartoon on the right is a very simple representation of how our molecules work. Bacteria is present, we'll inject our drugs. That drug will bind to the pathogen, presenting that sugar at high enough density to recruit those antibodies and drive rapid clearance of that bacteria. So since we closed our Series A in 2022, we, and by we I mean our CSO Helen Bright, has done a huge amount of work with the team to work through the mechanism of action of these molecules, really understand the immunology. And so what I'm describing here is even a simplistic version of that. And this is only describing the mechanism of the immunological arm of the molecule and not the direct acting antibacterial activity that we have coming from the binding moiety. So our molecules bind specifically to the target based on the targeting moiety. Once those alphamers, once that lead is bound to the target, that recruits these polyclonal antibodies. That leads to activation of complement, and that complement leads to assembly of the MAC complex, and it also leads to opsin phagocytosis. So we have multiple arms of the immune system coming to bear to kill bacteria. But this is the mechanism that exists regardless of what cell type we're going after to kill. So, as mentioned a couple of times now, the whole key to this platform is appropriate presentation of sugar to the immune system. And so when we design molecules, it's really thinking about how do we present those sugars in an appropriate density to recruit these anti-sugar antibodies. So when we are going after a cell which has a target that's expressed at high enough density, we can build molecules which have just a single sugar linked onto them, and that again leads to that sufficient density. If we are going after a cell which has lower expression of a target that we're interested in engaging, we can build multivalent molecules where we express multiple sugars at a time to ensure we've got that appropriate array. In terms of targeting moieties, we've exemplified everything from cyclic peptides, linear peptides, aptamers, Fab fragments, monoclonals, and small molecules. And we have that flexibility because of the IP that we have around those linkers, which is really what I would call the secret sauce. It really enables us to be thoughtful about how we engage the target. What's the right targeting moiety given the target, where it's expressed, how it's expressed, what's known, and then ensure that we can appropriately design the sugar end of the molecule so that we are recruiting those antibodies. So the two things that I want to point out on this differentiation slide is we have, as in the case of our lead, potential for building bifunctional molecules, which is great in terms of trying to manage emergence of resistance. And we really are focused on differentiated health outcomes. We often say if we're going to fail, we're going to fail big. And it's one reason why we spend a lot of time thinking about who these patients are, why they might be failing more traditional therapies, and thinking about when we consider moving through the clinic, how do we enrich a clinical study so that those patients are highly represented in our data sets, because we know those are the patients who are going to respond differently to a molecule like we're building compared to something that might be more traditional. And so just a bit of data. And what I'm showing here are a variety of different tool compounds, both in vitro on this slide and in vivo on the next. But these types of data, this type of data is quite representative of where we are with the data for our lead. So the figure on the left, what I'm showing here is decreasing compound concentration of a tool compound which is expressing alpha-gal. And you can see that at decreasing concentrations of drug, you have decreasing binding of anti-gal IgM. When you move on and look at what this bound anti-gal IgM is doing, we know that leads to complement deposition. And then we can tease out the effects of both the direct acting antibacterial activity that we see in the targeting part of the molecule, as well as the immune portion. So the gray bar is just our control, bacterial burden. When you add the tool compound CTX09 in with serum, so no antibodies are present, we can measure that antibacterial effect. And then when we add in our molecules plus serum plus those antibodies, we have a significant drop in bacterial burden that's coming from the immune component. And we see the same thing in vivo. So this is 2 different compounds in 2 different strains of E. coli in a murine thigh model. The first 2 columns in both of these figures, the closed circles and the open circles, are just our controls. When you've got anti-gal present but no drug, we wouldn't expect to see any effect on bacterial burden, and that's exactly what we see. When we add in the drug, but no antibodies, we can measure the direct acting antibacterial portion of the molecule. And then when we add in the antibodies, that's where we can measure the additional activity that's coming from the immune component. And again, this data is similar to what we're seeing with our lead. So our lead program, AVX01, as mentioned, going after gram-negative infections. Our targeting moiety does provide that intrinsic activity. We are bound to rhamnose rather than alpha-gal for this lead program. We've chosen a clinical candidate, so we're in that stage of very expensive drug development, which always makes my eyes water when I have to sign those e-checks. But it's very exciting to see how close we are to getting this compound into the clinic. So again, thank you to all of the people who've always been around the table to get us to this point. Our TPP is for HAP-VAP. We're going after pneumonia. So going after lung infections, we have data in animal models of lung infections demonstrating that we can get our drug into the lung. We do see activity there. So really excited to be able to pursue that, because when we think about the types of patients that we talk a lot about, lung infections come up a lot, whether we're talking about bacterial infections, viral infections, fungal infections. So we think of this not just as a really great first product to take into the clinic, but also gives us real validation that we could go after other infections in the lung and in these patients. So just in summary, I'm really proud of the work that our team has done to tackle a really hard question. I think everyone who's worked in the antibacterial space knows that this is hard for any variety of reasons. And the fact that we've been able to demonstrate both the direct acting activity of our molecules, as well as the immune activity of the molecules, and that we're so close to the clinic, I think really speaks to the excitement there is to use immunotherapy to address infections. And that's my presentation. Thank you.
Thank you very much, Jennifer, for this interesting insight. We directly come to our next speaker, which is Rida Mourtada. He is a co-founder and the CEO of Lytic in the USA. He studied in Toronto, Canada, and did his PhD in Harvard, MIT in the US. And his interest is in drug discovery, and in particular in stapled antimicrobial peptides and their antibody conjugates. And this is also the topic he will talk about today. So the stage is yours, Rida.
Thank you. Thank you, everybody, for joining me today to learn about Lytic's work in the antimicrobial space. So I like to usually start with, talking about antimicrobial peptides. This kind of compounds or natural products have been long known for now almost half a decade, if not more, since Michael Zasloff first isolated the meganines from the skin of the African clawed frog. And since then, we've discovered that, antimicrobial peptides is really an ancient version of an immune system that's, still used in insects, for example, in hemolymphs, as the first line of defense against invasion of infectious disease, such as bacteria, fungus, and viruses. And to date, we've discovered there's hundreds of these sequences out there with a whole variety of structures. Although the largest portion of this family is actually alpha helical in character. And one of the interesting things about this family of antimicrobials is that they all kind of work in the same sort of mechanism, in that they're able to fold themselves in a way that they become amphipathic. So they have a positively charged side and a greasy side. And in the presence of a bacterial membrane, what they're able to do is they're able to kind of sense the biophysics of that membrane, sense the charge, the fluidity, and they start to kind of slowly bind onto that membrane. And as they bind and aggregate, they start to porate. And once they porate, they lyse the membrane and the bacteria dies. What's fascinating about this is their ability to distinguish, between host cells and between the bacterial cells. And as I said, this has been found in all over nature. Interestingly, in humans, we've evolved our very complicated immune system. So our system doesn't rely as much on this ancient class of antimicrobials as much. However, we have found places where it seems like we still do use AMPs. For example, in sweat pores, turns out we do secrete something called dermacidin that can porate gram-positive bacteria and prevent them from, invading your pores. So it seems that overall, this family is highly utilized, and the big thing about them also, because of their promiscuous mechanism of action, it's very difficult for bacteria to develop resistance against them. And it's a very orthogonal mechanism to a lot of the things, or a lot of the molecules we currently utilize in the clinic to treat bacterial infections. Because here, they're not binding to a specific protein, they're not binding to a specific lipid, they're more recognizing the character of a bacterial membrane. And so it's been proven over and over, it takes a lot of evolutionary steps for bacteria to kind of change their membrane in order to avoid being lysed by antimicrobial peptides. However, and another interesting thing about that is, as they try to evolve, it seems that that also affects their ability in being infectious. And so even if they develop resistance, it seems like AMPs make them harmless then, because then the bacteria can't do the things it wanted to do because its membrane structure is so different. As I said, this class of compounds, huge potential. There's been a lot of interest in harnessing their ability to treat infections. However, as with anything that's a small peptide, they do have a lot of liabilities that have really made their translation, especially for systemic delivery, very difficult. And so, typical things is they tend to be low potency. So in nature, a lot of times these are secreted at the site they're needed. So they're concentrated within the site, so they're able to act. But if you were to, inject them systemically, you would need a lot of that peptide to be able to affect the bacteria. Second, especially for humans, as you're trying to adapt this, the selectivity, a lot of these natural compounds between human membranes and bacteria is not that great. So, hemolysis is one of the things people talk about a lot, but also there's, kidney damage and other aspects of this that makes it difficult. Third, they're not that stable. They are peptides. So they, if you inject them into our plasma, they'll be shredded to pieces. And lastly, because of all these liabilities and instability and poor PK, they tend to show low systemic in vivo efficacy, although there's a lot of people have shown you can use these as creams. And that's why, if you look at the clinical translation, a lot of people have focused on topical applications. The one intravenous application that tried to adapt the concept of an AMP into a small molecule was brilicidine, and unfortunately, that never made it past phase 2. So at Lytica, we kind of specialize in a kind of chemistry called peptide stapling. And what peptide stapling allows you to do is to introduce unnatural amino acids that have olefin side chains at the end of them. And when you use a ruthenium catalysis to do a metathesis reaction, you can basically link those side chains and form a brace around the peptide. What that does is it basically allows a peptide to fold into a helical conformation, especially if that peptide is meant to be in that conformation. And in the case of AMPs, based on all the biophysical studies people have shown, the bioactive structure of AMPs that allows them to engage, especially the helical ones, is being in that helical conformation. And so our thinking was, what if we lock them into that structure? Would that allow us to amplify their potency? Because now they're in the right conformation. They don't have to, fold in the wrong way. And furthermore, because now they're very well templated, you enhance their proteolytic stability. And so potentially you can turn this into a systemically delivered peptide. And so that's where we created the stapled antimicrobial peptide platform. And the goals we had was enhance their potency so that you can treat highly drug-resistant bacteria with low single-digit MICs. One of the problems I said is their MICs typically are double-digit, and our aim was to go for a single low-digit MIC between 1 to 2 micrograms per mil. Broaden the safety profile so that there's over 100x in selectivity between at least 2 log selectivity between red blood cell and bacterial cell. Since these compounds tend to be charged, ensure there's no kidney liability, and that's typically seen with polymyxins because of their positive charge and aminoglycosides. Make sure that they're highly stable and they show good pharmacokinetics, and then be able to treat infection via intravenous delivery. And in 2019, we published our first design principles for these stamps as we were trying to apply our technology to this class of peptides. And what we found as we were optimizing and screening these peptides is that it's really critical to think about the peptide in 3D structure and think about the biophysics of how you display the greasy parts of the peptide highlighted here and the positively charged parts of the peptide, such as the lysines. And if you're able to position these residues in 3D so that you can avoid having extremely greasy components across the peptide, you can then generate compounds that are extremely selective, especially for gram-negative bacteria. And so you can see here that we're able to lyse this bacteria around 2 micrograms per mil, but we don't see any hemolysis until you're well over 100 microgram per mil concentrations of the peptide. And so in the paper, we showed that once you introduce a staple, you can make AMPs extremely stable. And so in this example, we took a natural AMP, we put 2 staples on it, and now it's extremely stable against proteinase K. You can get really high safety profile, and at Lytica we've gone further and made this safe all the way up to 1,000. You can engender high selectivity down to even selecting between a gram-negative pathogen and a gram-positive pathogen. And as you optimize, we kept checking to make sure our mechanism of action is still the promiscuous lysis mechanism. And so we found that this mechanism does not induce resistance, unlike if you use a polymyxin, which relies on binding to lipids. You do not see resistance after co-culturing with low sub-MIC levels of the compound. And we also tested this against MDR clinical isolates, and we found that we have pretty strong activity against a broad range of critical gram-negative pathogens. Now, this was kind of the state of the art that we published in 2019, and we founded Lytica with the goal of taking this forward and creating, a real drug, ideally with better properties as well. And the profile we're going for is we wanted to create a stapled antimicrobial peptide that you can intravenously deliver to treat HAP/VAP. And ideally, we wanted to cover MDR Pseudomonas and MDR Acinetobacter baumannii and potentially a couple of gram-negatives, but really we were focused on A. baumannii and Pseudomonas as our main pathogens, because we saw, in terms of potential uses for this could be, of great use to patients with, chronic, infections and cystic fibrosis, and this could be of great use for, patients who have HAP/VAP And so we were kind of thinking about the market opportunity and the funding availability, and we felt that these kind of two pathogens offer enough economic incentives potentially to also lure in investors who want to invest in an antibiotic. So when we started, as I said, the big thing that we were trying to do is improve the potency, especially to cover as broad swath of clinical strains while maintaining the safety profile. So what you see on the left is we were screening a panel of 55 Pseudomonas strains that we got from the CDC's Antimicrobial Resistance Bank. It's an amazing service that is available to people in research, both in academia and in biotech. And what's great about it is the CDC is collecting these strains from around the country, and they actually have like the full panel of what antibiotics they're sensitive to, what they're resistant to. So it's a really good panel to test your compound against and to optimize against, because what we found, it's pretty reflective of broader resistances that you might see in the real world. And so you can see, the compound we started with, even though with the ATCC strains we were using, it was MIC of 2. Once we started testing against many more strains of Pseudomonas, we realized that actually our MIC is in the double digits, and there was much work to be done. However, our safety profile was pretty great because the IC50 in cell culture was closer to around 500 microgram per ml. So the goal here was, can we cover more strains of Pseudomonas, so get our MIC 90 to be at least a 2 while maintaining the safety profile. And so we started kind of screening different permutations of the sequences. So we played around with the amount of charge on it, and we found you can add a lot of charge, and sure, you will get more killing, but the problem with that is now you also start to shift your toxicity profile and start to lose on the safety part. We started to play around more with things like, okay, what if we, instead of adding too much charge, we tried to add like a little bit more grease onto it, and we got better than we were, but we were still away from our goal of an MIC 90 of closer to 2, but the safety profile was still maintained. And so as we were going about this exercise, we constantly found that you have to carefully thread this needle, because you're trying to give this peptide the ability to distinguish between very subtle differences between a bacterial membrane and a mammalian cell, but also you're trying to make sure it covers enough kinds of Pseudomonas, because as Pseudomonas grows in the world, we have some mucoid strains that make it difficult for the AMP to penetrate, and you have other strains that have been exposed to polymyxins, so their membranes are also a bit different. And so we're constantly tweaking around, and what we found in the end that you kind of had to also broaden your toolkit. So we started to explore non-natural amino acids, basically, to further allow us to thread this needle. And as we went further and further, we were like, oh, we're getting somewhere here. We're starting to find that as you tuned things with unnaturals, adding things like terbutals, maybe a fluorine here, you start to be able to get towards a safety profile that's acceptable, but also an MIC 90 where we got to the MIC 90 we wanted. We got MIC 90 of 2, while we got pretty close to the safety profile we started with. That was really exciting for us, because we have not seen this before with an alpha helical antimicrobial, especially something that can treat a broad swath of Pseudomonas strains. And in tow, what we found actually, that as we optimized against Pseudomonas, we also were optimizing against Abomani. And so you can see that actually the MIC 90 for Abomani was closer to 1 with our lead compound Lytic 496. And that was really cool to see that we were able to make the same gains on this different strain of bacteria and maintain a really good safety profile. And for context, these are large molecules. So when I'm saying, I have an MIC 90 of 1 or 2 micrograms per ml, if you want to go back to molarity, especially if you're trying to compare it to small molecules, Here you're talking about like 100 nanomolar potency, which is pretty remarkable for something that's purely acting on biophysics. There's no binding partner to it, so there's no protein it's binding to. It's purely recognizing charge, fluidity, and membrane structure. And I think that's something, we still at Lytica, have been working on understanding, especially now that we've pivoted to oncology, that membrane structure, there's a lot that could be discerned purely by these biophysical properties. So this is kind of the summary of what we found as we optimized. And as we were working towards getting the lead molecule series, we were also very cognizant of kidney damage, because like I said, this has been the Achilles heel of a lot of antimicrobials that do incorporate positive charge. And so we were pretty pleased that as we optimized up to Lytic's 496, we were able to maintain also a really great safety profile. And so, if we calculate a in vitro therapeutic index, you can see that there's, over 100-fold selectivity between Pseudomonas and a kidney cell, and more than 300 between kidney and Abomani. And so that was really exciting to see. And so then we took those learnings, and then we worked a lot on the in vivo aspects of it. Can we truly deliver a peptide intravenously? And what we found, and what we were kind of like ensuring, is the mechanism of rapid lysis was still there in our lead compound. And what was interesting, it was even there in vivo. So on the right, we were collecting lung samples around 4 hours post-administration of the peptide. And what was remarkable is, even at 4 hours post adding injecting the antimicrobial systemically, you could actually see that the CFU levels of the Pseudomonas in the lungs of the mouse have decreased from where they were. And so the kinetics of this was really incredibly fast, showing that going in vivo didn't change how these peptides behaved. They still acted in a lytic way. And we went and did a lot of deep dives trying to understand, did the peptide truly make it in there? And we even isolated the bowel from these lungs, which was a complicated procedure to get. But when we do bowel processes, we found that these bacteria were lysed. And then we were trying to figure out what's a sustainable dosing regimen so that we can get good responses overnight. And we found that what you would need is around 2 doses of this antimicrobial, and you're able to get a pretty sustainable suppression of Pseudomonas growth in these mouse models. Now, in terms of our goals as a company, I think we achieved what we wanted to get to, which was develop a lead molecule that showed the safety properties we were looking for, that was active systemically, and that was closer towards something that could be translated into an intravenously delivered antimicrobial peptide. However, I want to spend these last 2 minutes talking about like the other part of developing antibiotics, which is the funding part. And I think, for Lytica's case, we were originally funded through CARB-X, which is a wonderful organization that provided us with funding, with a lot of experts and a lot of people who were invested in helping us get through to the stage of lead series development. However, unfortunately, with the pandemic and cuts, CARB-X had to start to focus their portfolio, and so we were one of the companies that were let go out of the portfolio. But as we went to private investors to excite them about this, especially being a new class of antimicrobials, which is something we really need in the clinic, the excitement was kaput, because a lot of the investors in venture capital were basically like, this is all exciting science, but while you're solving a clinical need, there's a huge gaping hole, which is the economic parts of it. How can you make money with a novel antibiotic? And this is something I found that it's a big gaping hole at the end of a drug development that we really need more people to focus on and to talk about and to campaign on. You know, in the United States, there was the Pasteur Act, which is basically to allow, you know, a subscription-like model so that if you develop an antibiotic and it meets a significant need, you get, you know, a big payout at the end of it after you get your FDA approval, and then you get a consistent, you know, revenue stream to sustain the supply of that antibiotic. I know the United Kingdom has a program now that follows this. They call it the Netflix model. But we need more countries to enact stuff like that, because we need to be able to reward investors who are putting their capital at risk. You know, it still takes the same amount, a lot of money to develop a drug, whether you're in oncology or in antibiotics. And we need to be able to, you know, provide investors with that option. And so unfortunately for Lytica, we couldn't answer this problem, which is, you know, how will we recoup the investment? And so we had to pivot to oncology, where, you know, when I go fundraising now, it's like it's not even a question anymore of like, how will I make money once you get approved? That's assumed. And I really wish we have that in antibiotics, that we don't lose out on the innovation that's been happening in the pipeline, and we can restock our pipeline, because as we see in the news every day, you know, antibiotic resistance is a huge problem. And it's something that, you know, even in cancer, you know, a recent study came out showing that patients who get cell therapy and then, you know, pass away later on, more than 50% of the time, the reason they pass is not because the cell therapy didn't work, but because of the associated antibiotic resistant infections that they got that couldn't be treated. So it's a real problem, it's a real need, and I think we need to solve this issue scientifically, create novel chemical matter that can treat resistance, but also need to find an economic solution so that we can create a sustainable pipeline. And thank you. I look forward to questions.
So thank you, Rida, for this insight. Very interesting scientifically, and probably about economic models we will talk later. But now we go on to our third speaker of today, and this is Gregorio Iraola. He is co-founder and CEO of Kinzbio in Uruguay. He studied biology and bioinformatics. He did his PhD in computational microbiology, all of that at the Institute Pasteur in Uruguay. And he is an international fellow at the Wellcome Sanger Institute in UK. He is also adjunct international professor at the University Mayor in Chile. And his interest is in a One Health approach, integrating the human microbiome with its surroundings. Welcome, Gregorio.
Thank you for having me here. It's a pleasure to be speaking today about what we are doing at Kinzbio. So as the webinar was focused is focused on non-traditional antimicrobials, well, I want to tell you part of our story developing novel antimicrobials to both prevent, but also, and not least, treat bacterial infections using non-traditional phages. And that's let's see what this means in the following slides. You know, the phage field is has researched in the last decades with a lot of interest in the field to solve the problem of AMR and AMR infections. And indeed, we know that phages are the most abundant and the most genetically diverse entities in nature, because basically they are viruses that infect bacteria that are the second most abundant biological entities on Earth. But the truth is that only a small fraction of this phage diversity can be today isolated in the lab, and therefore used for industrial and biotechnological applications. This has been uncovered mainly due to metagenomic analysis that have revealed a vast diversity of previously unknown phages in different environments. So we know that there is a gap between what is out there in terms of phage diversity and those phages that we are using for biotechnological, therapeutic, and other applications. And this reminds me what happened a couple of years ago with the human microbiome field. You know that the human microbiome field has surged in the last years with an extreme growth curve. The publications and also the applications regarding the manipulation and the interventions on the human microbiome have been growing a lot. Indeed, there are a couple of drugs that have been recently released based on human gut bacteria, on live bacteria. But the truth is that until 2016, more or less, we were able to culture a very small fraction of the human gut microbiota. And the paradigm in that moment was that basically the majority of the human gut bacteria were unculturable. And one single change in the methods that were or started to be used to isolate bacteria from human gut samples changed everything. Just the introduction of a single culture medium to the standard pipeline to culture bacteria from the gut allowed to uncover a bigger share of culturable bacteria that basically started to open the field to, of course, better understanding, more elements to be used to manipulate and to redirect the functions of the human gut. And so on, we have started to discover more and more culturable bacterial strains from the human gut. But I'm talking about bacteria here because it's a very interesting parallelism what is happening today with phages. These methodological barriers that were there for bacteria and were overcome allow basically today to have a lot of clinical trials ongoing with several of these bacteria as therapeutic assets that were previously unable to be cultured. So with phages, I think it's happening the same. Today we have a methodological barrier that only allows us to use a very small fraction of phage diversity that is present in nature. So this is basically preventing us to exploit phages in their full potential for different therapeutic applications. I was saying that these methodological barriers allows us just to culture and to use a very small fraction of the phage diversity that is present in nature. Indeed, this diversity is sorry that this is moving very slowly. Sorry for that. I'm not being able to move my slides.
Okay. So,
okay.
If you just take next slide, I'll move on.
Yeah. Okay. Thank you, Victor. So this diversity that I am talking about is basically, I presume, summarize. When we consider the genome size of phages, we can categorize phages in different genome size categories. For example, typically a phage is around 50 kb in their genome size, but we have phages that are much bigger than that, including what we call jumbo phages, that are phages with over 200 kbs in genome size. But if we see the distribution of, for example, public phage genomes, the NCBI, we see, for example, jumbo phages and larger phages are very the distribution is very biased towards regular phages. So the jumbo phages and bigger phages are very poorly represented. And this is the same in most biobanks from companies, from research groups. Most pages that we are using are regular size pages with around 50 kbs. So this basically is what we are then using to develop therapeutics, to develop other applications based on pages. Next, please. Okay, it's there. So as I was saying, phage isolation methods only recover small size genome phages. So this represents a bias and a barrier, a methodological barrier to fully exploit their potential. Phages enclosing bigger genomes have for the bigger genome sizes have expanded genetic and metabolic repertoires, but these phages, as I was saying, are rarely recovered with the standard methods we use in routine analysis to recover phages from nature. So this basically is the problem and the key here. The methods we historically use, the lab methods we historically use to recover phages from nature, only catch a very small fraction of the actual genetic and genomic diversity that exists in nature. Next, please. So at Kinzbio, we have basically overcome this methodological barrier by developing our own methods and protocols that allow an enhanced discovery of phages from natural samples, with particular emphasis on phages with bigger genomes that we call jumbo phages. These methods, when we apply them to different sample types, have allowed us to discover previously unknown species of big genome phages that we call jumbo phages. And we have also developed additional methods for improved genomic analysis of these phages. Also, these phages not necessarily behave as the standard phages in terms of their propagation and manufacturing characteristics. So we have also created a proprietary set of tools that allow us to work with these rare and bigger phages. So part of Kinzbio's proprietary package, an IP package, is of course a biobank made of these unique phages that offer some advantages that I will tell you later. And also we have developed software and other accessory tools that allow us to better characterize the biology of these phages and optimize their applications for preventing and treating infections. So why it's important, or why we consider these phages are interesting and it worth to recover and work with these phages that are vastly overlooked. First, these phages are almost exclusively virulent, so this basically minimizes the chance of integration of the phage to the bacterial genome. So for safety reasons, this is important. We say that these phages are resilient because their expanded genomic repertoire and metabolic potential basically includes a lot of mechanisms that these phages use to basically fight against the anti-phage mechanisms that bacteria usually encode, typically CRISPR-Cas, RNA systems, etc. So it's more difficult for bacteria to develop resistance against these huge phages, because these phages have their own ways to bypass these immune systems that bacteria have evolved to kill or to prevent phage infection. One of the most important aspects of these phages is that they are typically broad spectrum phages. The phage infection paradigm tell us that basically one single phage can kill a very narrow set of strains within a species, or a very restricted diversity of serotypes or whatever you want to classify within a species. And this is a problem for the development of phage products, because typically you need to combine different phages to reach a broad enough antimicrobial activity against a target bacterial species, target pathogen. So the broader activity of jumbo-phages allows to develop more minimalist products that can consist in a very small number of different phages that can even target more than one bacterial species that one wants to kill. Also, they are very resistant to environmental stress and, for example, pH, for example, temperature, and other perturbations. So this makes them very interesting to deliver these phages in the environment for prevention. For example, this is something I will go deeper in a couple of slides. And also, this improves, for example, shelf life of products that contain these phages. Another interesting characteristic is, and this is something we are exploring with our proprietary software for genomic characterization powered by artificial intelligence, is that these bigger genomes encode a very vast diversity of unknown genes that probably enclose new enzymes, new antibacterial mechanisms, or new, for example, DNA editing enzymes. So most enzymes that are currently used in molecular biology as standard tools are derived originally from phage genomes. So phages enclose a high potential for the discovery of novel enzymes and novel molecular mechanisms that can be used in diverse biotechnology industries in the future. So it's something that we have there in a side, but there is a possibility of exploring this further. And also, as they have bigger genomes, in case one wants to perform genetic modifications of these phages, for example, these phages can tolerate bigger genetic cargoes because their genomes are bigger. So this is why we are basically focusing on exploring the biology of these phages and turning this research into applications for treating and preventing AMR infections. Next, please. as I was saying, Kinzbio core technology is focused and based on these things that I was telling you before. And with this core technology, we are developing two main programs. One is focused on personalized medicines, and in this sense, we have developed a personalized phage therapy platform that is currently being applied. We have already treated patients successfully with our technology, and we have started an early commercial stage in which we are commercializing phage therapies in Uruguay and in Latin America to treat different antibiotic-resistant infections in both critical and chronic patients. And on the other hand, what we are developing through partnerships with the global leaders and global leader companies and other organizations is an application for infection prevention and control that is basically based on the delivery of these very particular phages on hospital and domestic surfaces to prevent and remove contamination with hard to clean and hard to remove AMR bacteria. Next, please. So I want to tell you one example of these two main applications we are currently developing. This is the case of the first patient we treated with our technology. This was a cancer patient that was admitted to an ICU due to recurrence of its cancer condition, and within the ICU, the patient got colonized with Pseudomonas in the lung, and after more than 6 months of being there, this Pseudomonas was unable to be clean with the standard antibiotic therapy, and at the end, the bacteria had evolved resistance to any available antibiotic. So we started a 12-day nebulized phage therapy that at the 4th day after treatment started to show negative cultures for the Pseudomonas, and 1 month after the treatment was ended, the patient was discharged from the ICU and discharged from hospital. So it was the first time phage therapy was performed in Uruguay, and also in many of the Latin American countries, at least in the last decades. So this basically generated a lot of demand and a lot of other cases that started to reach to us, and today we are receiving more or less 1 request per week, and we are moving ahead with more treatments. So this has been really fascinating and an exciting journey that started just a couple of months ago, because the first treatment was this year. So this motivated us to move ahead with this model that basically is going to be developing 2 ways of administer phages in Latin America and also in other parts of the world, because we have started to talk with a company in Spain that wants to bring these to Europe. So it's a possibility also to start scaling to Europe in the near future. But we are starting a process of installing different phage therapy units in different Latin American countries. We are starting to work with a big hospital in Sao Paulo, in Brazil, as the first steps outside Uruguay to establish phage therapy in this hospital, but also we can offer phage therapy from Uruguay to patients that can travel to the country and can be treated here locally. So I have a lot of more things to say about the model and how we are planning to expand and scale the use of this in different countries, but for time constraints, I will move to the other case example that I want to share with you. So next slide, please. As I anticipated in a couple of slides ago, the other application we are developing is to prevent infections through the intervention with phages over abiotic surfaces. You know that the built environment has its own microbiome, including harmful pathogens, and this is particularly relevant in high antibiotic pressure settings like hospitals, where a lot of AMR bacteria are deposited on surfaces, and this can end in patients causing very, very difficult to treat infections. So our aim is to cut the spread of this bacteria before patients are infected as a complementary tool to the standard IPC methods that are today used to prevent this. That it's evident that they are not fully working because the transmission of bacteria from surfaces to or from the environment to the patients is something that is relevant in infection control and prevention. So basically, we can produce aerosolized or atomized phage preparations that can be applied over surfaces, and the phages can resist in these surfaces for considerably long periods of time, preventing and removing certain bacteria that we want to remove from these surfaces, but without affecting the other bacteria that could be beneficial that are part of the environment. Next, please. In this sense, we have performed different experiments in vitro, and also we have performed field trials inside intensive care units in hospitals in Uruguay. And basically, to summarize this, we have seen that our phages can effectively remove bacteria from experimentally contaminated surfaces, different surfaces, and also these phages can be effectively recovered from these surfaces a lot of hours after the application was performed. Also, we have confirmed that our phages are resistant to chemicals that are commonly used in typical disinfectants, and this supports their addition to, for example, surface protection formulations, and are compatible with the treatment of surfaces with other chemicals that are used in IPC. And the most important, I think, is that we have also confirmed this in situ in the intensive care units. We have performed these phage atomizations in isolation boxes during the state of colonized and infected patients. Before this, we have confirmed that the bacteria that were infecting these patients were indeed present in the surrounding surfaces and environment of that are part of the isolation box. And after treating these surfaces with our phages, we have observed a significant decrease of the bacterial load a few hours after the treatment were performed, and this effect was retained several hours after, meaning that the phages can also prevent the regrowth of this bacteria. And basically now we have been started the process of registration of this application here in Uruguay, and we are close to obtain the clearance to start applying this more massively in different hospitals, and we are also partnering with different organizations to move this into its next step, hopefully in the next few years. We are independently of this. We are also partnering with a multinational company that wants to develop a new generation of surface protection products for the domestic environment to prevent, for example, foodborne pathogens and pathogens that cause urinary tract infections, etc. So this is basically how we are doing this, where we are and where we go. So just one slide remaining. Next, please. This basically summarized what I have been sharing with you today. As final remarks, we have that basically Kinzbio have developed a new phage technology platform that is able to, we basically with this platform we are able to expand the diversity of culturable phages, and we are using these bigger and more diverse phages as antimicrobial tools. Based on these biobank of phages, as I told you, Kinzbio has successfully performed personalized treatments that have achieved complete eradication of the infecting strains for different top priority pathogens, including Pseudomonas, Klebsiella, Acinetobacter, Staph aureus, etc. As I mentioned, this is now a new therapeutic option that is already commercially available in Uruguay, and hopefully in the next few years it will be expanded to the rest of the continent and also internationally. And the last two things, the ongoing partnership we have with this multinational company is also a long-term project of 5 to 7 years that has the aim to put in the market a new generation of these phage-based disinfectants for domestic environments. And there is another partnership under consideration to keep developing the hospital infection prevention control based on phages that we are also exploring, and hopefully we can move forward with this as well. So that's all, and thank you very much for your time and for having me today.
Thank you very much, Gregorio, for this interesting insight. We had now 3 talks with non-traditional antimicrobial strategies, and we have now time for some questions. And since we have quite some questions, I would like to divide them in more scientific questions and in questions about economical models or regulatory. And I would give just a few minutes to the scientific questions. And I'd like to ask you to answer quite to the point so that we can move quickly to the economic questions. And the first question goes to Rida. The question is, what is the route of administration you used for the Lytic 496 in the lung model? And do you know different routes and if they affect the PK/PD model? And do you see in vivo toxicity or immunological responses in your therapy?
Okay, that's quite a bit of questions in one. So basically, we had to inject the animals intraperitoneally to deliver it systemically. We found, one of the issues with mice is what rodents in general is they're very sensitive to positive charges. So it's well known with polymyxins that if you inject rodents intravenously rapidly, they develop kind of a histamine response to it. And we found with antimicrobial peptides, it's a similar thing. So we have to inject them slowly, but unfortunately, you can't see. When we did our studies in rat, we used an IV pump, so we could slowly infuse. But in mice, we found it's very difficult. You need to use subcutaneous inlet pumps to slowly infuse. And so we found that the absorption kinetics from the peritoneal cavity was good enough to give slow plasma entry of the peptide, and you reach a Cmax. And in general, we found that the kinetics are fast for clearance with the peptides. So we had to do some formulatory work to make sure that the clearance is not too fast for these peptides. So we definitely had to do quite a bit of in vivo optimization. I think the last thing was, did we see any immuno, I guess, autoimmune, like ADAs, any drug antibodies? We didn't see any immunogenicity, but the issue is all these models are really quick, because like these infection models are like a few days. We didn't do long-term studies, and from our discussions with experts, everybody said that the in vivo models are not that predictive for humans, and you really just look for those in phase 1 trials, especially since our peptides have unnatural amino acids. So we couldn't even use like immuno software that could predict immunogenicity.
Okay, thanks a lot. We have another question for Jennifer. How specific are the alpha-meres to different pathogens is one, and also how do you control the immune response in terms of overreaction in already challenged patients?
I'll start with the second question first. If Helen was on the phone, what she would tell you is that the complement pathway, for example, is one that's very tightly controlled and doesn't have a positive feedback loop associated with it. So we imagine that anticipate that there will be quite tight regulation of the response. In our animal models, in our early tox work, we've seen no suggestion of a hyperimmune response, any sort of cytokine storm. But of course, that's something that we'll be testing further and certainly looking for as we move into the clinic. From the first question, in terms of specificity, we're targeting LPS, so that provides a level of specificity for those gram-negative bacteria.
Okay, thanks a lot. And we have another scientific question for Gregorio. The jumbophages you said have a broad spectrum, which seems very unusual. And the question is, do you understand the mechanism? Do the phages bind directly to the bacterial membranes or to some other molecule on the cell surface? And is that conserved across species?
The question about the mechanisms. We understand some of the mechanisms, not all about them, because indeed there are very few studies in the past about jumbophages, and we have performed some of them. But what we have seen, for example, is that jumbophages code for multiple versions of bacterial cell receptors. So, for example, they have multiple variants of genes coding for proteins that are recognizing cell surface receptors. So we hypothesize that in certain way the phage progeny can recombine and express different variants of the fiber tail proteins, for example, that are those in charge of recognizing the cell. So with this, the phage can play around and basically this is one of the explanations we have. But a lot of these things we are seeing them just on the experiments, and we don't yet understand the mechanism that are underlying these observations.
Okay, thanks. So now we have quite some questions which tackle more or less the same topic, and this is what are the costs of goods of your innovations, and how can this intervention or these therapies be upscaled for a broad use, which is a bit in the direction what could be an economic model to develop such strategies, such innovative technologies. Maybe Rida, you want to start?
Sure. So, I mean, this was a question we got a lot, especially when we got our CARB-X funding of like, how are you going to make sure you're able to scale this and make it not so expensive? And I think, thanks to the GLP-1s, peptide production is not going to be an issue for the world because now there's so much demand for peptide drugs. We're going to see probably cost of goods of synthetically made peptides going down, given how much global capacity we're building for GLP-1s. And our peptides are solid phase. So I think the typical course of the compounds is not going to be more than several thousands of dollars. I think the issue lies in that what you're competing against generic antibiotics in the marketplace, which are a few bucks. And if you're a brand new drug and you want to recoup the cost of the investment, it's very difficult to recoup it when you're dealing with very cheap generic drugs. And also you're dealing with the issue of stewardship, where you're going to be used only in very narrow settings. So you have very few patients, and then now you can charge very little. You can't charge something like a cancer drug anymore because insurance providers won't pay for it. So it makes recouping the cost really difficult, and that's why we saw, Achaogen go bankrupt after Zemdry got, approved within like 7 months, and they weren't charging an insane amount of money, but they couldn't keep up because. And I think Ryan Sears has a really good talk series about the cost of commercializing a drug, which is something people don't think about, of keeping your drug supply chain going. It's a very costly thing. And if you can't meet that, you can't maintain the supply, even if you have the approval and all. And I think that's why you need a subscription or some sort of guaranteed funding to keep the supply chain moving, or else it's going to be very difficult.
Okay, thank you very much. I'm afraid we have to finish at this point. Thank you very much to the speakers for these excellent talks and these insights in your strategies. And thanks also for the audience. I hope you had all an interesting afternoon. And with that, I would like to hand over to Victor again.
Great. Thanks so much, Sina, for moderating today's webinar. And thank you to the speakers for your presentations. So next in the webinar series, we'll have a webinar on the 19th of September, which will be an introduction to antibiotic research and development. So this is intended to provide an overview of the field from discovery to clinical trials and also pharmaceutical development. And it's aimed at all those with an interest in the field, no matter their level of expertise. So please do invite all of your network to join. You can find further details on this already on the Revive website. So please also make sure to keep an eye out for our newsletters and follow GARDP on X and LinkedIn to hear the latest updates from Revive. So that will be all from us. Thanks for joining today's discussion. I hope you found the webinar interesting and useful, and that you'll join us again in future. Please also encourage your colleagues to join as well. Please fill in a survey afterwards to inform us on how to improve these webinars. Thanks again and goodbye.